Good afternoon. Thank you for joining us for the virtual 9th Annual Forensic Science Symposium Lunch and Learn Poster Session, hosted by the Global Forensic and Justice Center. I'm Becky Carter, your moderator for this poster session. I am happy to introduce Dr. Nicholas Scott, Principal Scientist at Riverside Research, with his presentation, Bloodstain Detection on Camouflage Clothing Using Machine Learning Analysis. Welcome, Dr. Scott.
Good afternoon, everybody. The name of this presentation is Bloodstain Detection on Camouflage Clothing Using Machine Learning Analysis of Noisy Hyperspectral Imagery. This was work that was done in collaboration with my colleague, Sarah Jensen at Services Food. I'm, again, Dr. Nicholas V. Scott. I'm Principal Scientist at Riverside Research Open Innovation Center. My background is geointelligence, but I'm traditionally trained in physical oceanography, so I have a PhD in physical oceanography, but I transitioned into intelligence, and it turns out that many of the skills that you use in intelligence are somewhat transferable to forensics. So I'm giving talks today from an intelligence perspective and trying to apply that to forensics to give you a different perspective on some of the problems that you may be facing. Just would like to give an introduction of what I'm going to cover in this talk, or basically the guts of this talk. Military, law enforcement, and intelligence agencies are often need to quickly assess the presence of wounds on bodies in remote positions. Hyperspectral imagery, a proven technology for the detection of anomalous substances in heterogeneous environments, has seen use as a photonic detection tool for the wounded. This is mainly, but not exclusively, in the area of bloodstain imaging of bloodstained clothing. Blood on clothing often appears in different configurations, which change due to stresses that smear the fluid along the material. If a law enforcement person gets shot or a soldier gets wounded, as the soldier moves back and forth on the ground, the blood tends to smear on the clothing, and that's basically the problem that we're trying to assess. The detection of this modulation is important in assessing HSI and multispectral imagery technology versatility in bloodstain detection on clothing. Again, we're trying to attack or address this problem of how can HSI and multispectral technology, which is basically a crude form of HSI, with respect to the problem of bloodstain detection on clothing. Blood detection under conditions of smearing on camouflage clothing is explored using HSI machine learning algorithms. Image segmentation for different blood smear conditions is performed, and this is performed using machine learning algorithms. This is particularly non-negative matrix factorization, competitive leaky learning, and Laplacian Eigenmap-based anomaly detection. HSI characterization of bloodstains on clothing, again, leads to inexpensive MSI technology capable of performing remote sensing-based bloodstain image segmentation. That's what we're really after in the future. The experimental setup is as follows. A 10-inch square digital camouflage cloth was placed inside a light diffuser box illuminated with a pseudo-sunlight spectral light source, as you can see on the right-hand side. A Resonon Pika-L hyperspectral imagery camera capable of capturing spectral imagery measurements was used. In particular, it was used for cloth with deer blood and cloth without deer blood. Again, I want to make it clear that we're not using real human blood. We're using deer blood as a proxy for obvious reasons. The specs for the camera are shown at the bottom, which you can read at your leisure. Average spectral profiles of the clean cloth and the bloodstained cloth were taken, as shown basically in the top part of the figure. Spectral amplitudes for bloodstained cloth are, on the whole, lower than for the clean cloth, and that's because the reflectivity of bloodstained cloth is lower than for clean cloth. Spectral signatures for both blood smeared cloth and clean cloth have distinctive peaks at 436 nanometers, 547 nanometers, 612 nanometers, and 709 nanometers. these are related to both the true physical reflectance, that means the photons actually coming off the cloth, and also the type of white light spectrum that also influences the peaks that you see. So it's a convolution of both. On the other hand, bloodstained cloth has small spectrum minima and maxima between 420 to 550 nanometers and 620 to 700 nanometers. This is essentially due to the bloodstained cloth photonic structure. So we consider those bumps that you can see between 400 and approximately 500 are definitely due to basically the blood stain, the blood on the cloth itself. What you see in this image are the RGB images of the blood smear targets themselves. Again, we're dealing with 3 blood smear targets where the blood smear is increasing from left to right. I want to make it clear that the total mass of blood that we're using in each of the images is the same. The only thing that's changed is its distribution on the cloth. So I'm taking a knife and I'm smearing the blood into the fabric as you go from blood smear 1 to 2 to 3. 3 HSI cubes of blood smear cloth targets were taken with increasing amounts of blood smear appearing from left to right. And the image chips, those are the white boxes, were actually extracted for further analysis. Others, the specs for the HSI camera that were actually used in the experiment are in the bottom, and you can also read that at your leisure. It's not that important for the purposes of this talk. The first machine learning algorithm that we use is a very cute technique called non-negative matrix factorization. Again, what you're seeing is the results of the non-negative matrix factorization for different blood smear conditions, blood smear 1, 2, and 3, again, where the smear is increasing. Non-negative matrix factorization, in a nutshell, decomposes the HSI data into 2 different matrices, an eigenvector matrix and an abundance matrix. And what you're seeing basically at the top are the abundance matrices for the different blood smear targets. Just basically one slice from the array of abundance values that you get from the NMF analysis. The NMF, and what's important about this, what is important about what you see is that the NMF analysis is able to segment the blood-stained cloth and the clean cloth very clearly for the first 2 targets. So if you look basically in target 1, you'll see 2 heavily blue spots. That's the blood stain. So very crisp segmentation in that image. In the next image, again, you see these blobs of blue, which are basically speckled across the image. Again, that's the blood. The red is the camouflage, which you can actually see because it has these very crisp lines. And then in the third target, you see basically faint signs of blood, but it's very, very faint. You cannot really see a very strong signature of blood. Again, decreasing segmentation power in the third target, which has a lot of blood smear, but the first 2 targets, really pretty crisp, very crisp image segmentation. In particular, in the blood smear target number 2, you see like a little blob on top of the red camouflage. That's definitively the blood, with the white dot being the reflection of light from the inside, from the light source. The next machine learning algorithm that we use is competitive leaky learning. Again, same blood smear targets. Competitive leaky learning is a form of clustering, neural network based, with a similarity between input spectral signatures are calculated numerically. So basically, what you're doing in each of the images that you see, you can think of basically each of the pixels having basically a lot of energy in wavelength from 300 to 1050 nanometers. What you're doing, you're segmenting or grouping bloodstain signatures with bloodstain signatures and camouflage signatures with camouflage signatures. So you want to group like pixels with like pixels. And so we were able to use 4 groups to really distinguish the blood-stained cloth from the camouflage cloth. And what you see in black is the blood-stained cloth. Again, you can see the increasing amounts of smear as you go from blood smear 1 to blood smear 2 to blood smear 3. There's a small decrease in segmentation power in blood smear 3, but still you can see a large amount of segmentation. SIA competitive leaky learning analysis again was able to segment the bloodstained cloth from the clean cloth for the first 2 targets, slight decrease in segmentation power in the 3rd target. We can take further smaller image chips from the targets to use the next algorithm, which is called Laplacian eigenmaps. Laplacian eigenmap is a form of analysis that's data-driven, where which is a local preservation mapping that uncovers anomalous structures in the HSI data. So basically, you can think of Laplacian eigenmap as a form of goggles that you put, a mathematical form of goggles that you put over your eyes that finds local anomalous structures that don't seem to belong. Since blood in this situation is anomalous, it tends to focus on that. And what you see in blood smear 1, blood smear 2, blood smear 3, that the blood is cleanly segregated from the clean cloth. So the blood-stained cloth, which is shown in red, is cleanly differentiated from the camouflage cloth, which you can see in yellow and light orange. And you can see that for targets 1, 2, 3. Again, increasing blood smear as you go from left to right. The last machine learning technique that we use in our analysis of blood stains on camouflage cloth was a distance metric analysis. So basically what that is, if you have a blood stain area on a camouflage piece of cloth, that implies the image, if that blood stain area goes up, the image segmentation power goes down. What that means is that it's a philosophical thing and it's a signal processing thing. One can only tell if you have a blood stain by what is not blood stain. So that the more that you stain your cloth, the less likely you're going to be able to differentiate it. It's as if you look at a blood stain too closely, you won't be able to see it. For a good example is this: an ant on the blood stain would not be able to really see or sense segmentation, but a human who's looking at the blood stain and a clean cloth from a distance can discern it, because there's a difference between the blood-stained cloth and the clean cloth. We can see it. The ant can't. So as the blood-stained area increases, the image segmentation power decreases, which causes confusion between the camouflage cloth and the blood-stained cloth. So the distance metrics are calculated as a measure of distinctness between clean cloth and blood-stained cloth. In particular, we use 2 very strong covariance-based distance metrics: the Kullback-Leibler divergence and the Bhattacharya distance, both distance metrics which are used in this, the machine learning field all the time. Bhattacharya is actually still alive. Really, really nice guy. Large values of the KLD and the BD really imply strong separation of clean cloth and the blood-stained cloth. So again, which we want that strong separation to be able to do the image segmentation, and the large values for the KLD and the BD are indicative of that. So these are basically the results of the Kullback-Leibler divergence and a Bhattacharya distance. Again, large values for the distance metrics means improved blood-stained clean cloth segmentation. The distance metrics were large when the blood smearing was small. So if you look on the left-hand axis, you see small, medium, large. That's the stain size. We see a peak in the KLD and the BD when the blood stain is small. We also see a peak in the KLD and BD when the number of principal components is also small. So basically what that's saying is the distance metric is largest when the blood smearing is small. That means, again, your blood stain size is not too big. And when you're using a small number of principal components, that means a small number of the strongest components from the original physical spectral bands of the HSI. You can really, for practical purposes, that's saying you can really segment blood if your blood stain is not too big with respect to your field of view, and if the number of principal components, your strongest components, are small. So the conclusions are that machine learning-based HSI is possible but its efficacy decreases as blood stain smear increases in surface area. Small PC numbers associated with high distance metric values suggest that MSI technology for blood smear detection is possible. And this is good for MSI tech because it relies on a small number of spectral bands. Again, you can see the MSI tech on the right-hand side. It uses a smaller number of spectral bands in the HSI, and that's good for us because we just were able to show that the distance metrics like small numbers of spectral bands or PC numbers. For the first point, the machine learning detection, blood stain detection is possible and its efficacy decreases. What that means is that we think or we believe that if a soldier or law enforcement person gets hit in the leg or on any part of his body, as he rolls on the ground and smears the blood, detection is going to get harder. So relative to your field of view, you don't want your blood stain taking up too much area. Of course, we can't control that, but we can say that the efficacy of this technique of detecting blood stains on clothing is going to decrease. This is the original poster as I had it. Again, the points that I just talked about on the left-hand side. I want to thank you for taking time to listen to this presentation. I'll take any questions.
Dr. Scott, based on what you presented, the CLL spectra segmentation looks to be very impressive. Do you have an estimation on the percentage of surface area covered by bloodstain versus uncovered by bloodstain that would optimize the results from this type of imaging?
Actually, very good question. This is just the preliminary stage. I would like to have my intern actually calculate that to rigorously show that basically what you're indicating, that there are robust trends as the blood smear basically increases. So it's a very good point. That's the next step to take. This is just, again, the preliminary stage. Thank you.
Thank you. That's very helpful. It's interesting to see where this research may be going later.
Right. I just wanted to basically give a prequel to some of where this research actually started. Basically, how did all this work start, and why are we using laboratory conditions instead of basically doing capturing imagery basically in the field? And so I want to just illustrate or just talk about a few points. Military-based agencies. I support the Air Force, so a lot of but this is totally pertinent to other military agencies. They operate in areas of contention, and they're often tasked with search and rescue operations in high altitude remote sensing areas. And HSI has seen some use in search and rescue of the wounded and the bleeding. It's actually been a very useful tool, basically in the Middle East. That's all I can really say about that. If improved sensitivity of blood smear on camouflage clothing is desired, how can machine learning aid in this? And so we tried to address this in a preliminary fashion. Preliminary experiments were performed to assess the degree to which HSI and machine learning can detect increased changes of blood abundance on camouflage clothing. And the question is there a minimal amount of blood contamination on camouflage clothing below which detection is impossible? And it has implications for detecting the wounded in hostile environments, including urban areas. Miami and Florida, you guys are bombarded with high glare, basically light scattering, you're in a high glare environment. So there's a need to basically find robust methods of pulling out information in those environments where you have a lot of reflective glare. Miami, which I've been to many times, is nothing but reflective glare. So this is where this work started from. So the initial experimental setup is what you see is a 60 by 36-inch cotton polyester camouflage sheet was laid out on an asphalt parking lot and it was held down by steel plates. And we dripped cloud blood on the sheet in increasing amounts and then took HSI cube samples after each blood injection. And you can see basically the blood on the camouflage cloth in both images. Here would basically the experimental setup where we have a linear distance between the camera lens and the target of 25 feet, 45-degree angle of descent, or 45 degrees below the horizontal, and the ambient light conditions were sunny skies with a temperature of 53 degrees. As you can see, basically what you see on the right-hand side are two images of the camouflage cloth with blood stains. And you can see the blood stain in the lower right-hand corner. And the first image has a small amount of blood stain. The second image, a large amount. Light scattering noise, as you can see, is a very big problem. You can see the fuzziness in the imagery. The light glare prevented clear segmentation of the blood stains on the clean cloth. So that glare was a big problem and was impeding detecting the blood-stained cloth. So the glare noise prompted investigation of polarization for noise reduction. Basically, it was the impetus that got us to move into the laboratory. So again, the second part of this research was born from the failure of the first. Again, I want to make it clear that oftentimes science doesn't work. If you have an idea and the first time around, it doesn't work out very well, but you pick up the pieces, you think about what you did wrong, and you try to move forward, trying to basically salvage that which you had from the first. So the preliminary project, the first thing I talked about was a way to salvage this work that we tried to do, but didn't work out very well.
Thank you, Dr. Scott. That actually answers a question that I had in my mind. My background is in photography, and I know that glare off of reflective or wet surfaces often impedes the ability to record detail and would then, of course, impede the ability for analysis, whether visual or computer-based. Have you seen significant differences, or actually have you tested significant differences between wet and dried blood stains?
Actually, that's related to a presentation I'm giving tomorrow, this idea of wet and dry. We we have investigated that phenomenon, but not for blood. There are substances that basically transform or modulate the electromagnetic field or the photons which are being reflected. And so there are techniques, and there has been an experiment that we use. It wasn't for blood, but there was an experiment that we did to really try to address that problem, using which I believe you're probably familiar with polarization. Photographers such as yourself use polarization lens to really basically get a very clear image so that you can do photoshopping or what have you. That was something that we would like to do for this experiment as a follow-on, but this again was the first shot at this problem using a laboratory setting, but definitely polarization, and which is very sensitive to wet and dry surfaces, is a very important thing to address. And that idea of wet and dry and the role of polarization and getting better scene contrast is actually related to something that I'm talking about tomorrow. So yes, very good question.
Well, I definitely don't want to have any spoilers for your presentation later.
No
To
problem.
all of our participants who may have just joined us, please make sure that you enter your questions into the Zoom chat window. I will ask them of our presenter, and anything that you think of later on, you can certainly email to forensics@fiu.edu, and we can forward that on to Dr. Scott for his answers. I am very familiar with polarization filtering from a visual perspective, and so it's fascinating to me to see how that affects the machine learning analysis on this imagery, especially since the point of camouflage clothing is to camouflage. It's a visually dense
Correct.
surface and substrate.
Correct. We wanted to, that's the reason people may be wondering why camouflage was used. The reason is that we use camouflage for the reason that you just indicated. We wanted a very heterogeneous background that can potentially confuse the algorithm. So giving this highly heterogeneous background, can we still pull out trace amounts of blood on the cloth? That was really what you hit on. It was actually the reason why we use camouflage clothing. It actually also benefits, of course, the military, but it was really a physics thing that we want like the worst background possible for blood detection. And, we wanted to try this to see what would happen. It's a cotton polyester mix. So mixing the blood into the cloth was not easy because it naturally wants to repel the liquid, but we kept pressing it into the material until we were successful. And again, what we're able to show, and you can see it in this poster, that as you go from left to right, that's the top column in the middle row of this poster. As you go from left to right, when you get to the highly blood smeared conditions, a lot of algorithms, at least the ones that I use, have a hard time distinguishing between the camouflage pattern and the blood stained pattern. So it gets confused. Again, machine learning isn't perfect. And that's another thing that I wanted to let people know, that these algorithms that we use, I work in the machine learning lab at Riverside Research, they're powerful, but they're not perfect. And there are many situations that they can pull out really, really interesting and important features, but they do fail sometimes. And we want to basically show the conditions when it does well and when it fails. And as it turns out, with increasing blood smear, it tends to fail. Again, I don't have the robust numbers to give you, but you can at least see in the distance metric that there's a trend that suggests when you have good image segmentation power and when you do not. That's shown on the right-hand column, basically the two figures, A and B, which are in yellow, light blue, and dark blue.
It's fascinating to see machine learning algorithms applied to this type of visual analysis. And it's also pretty clear to me, at least, again, as a layperson, that the less background pattern, I guess, you have or sample relative to the discrepancy or the blood coverage, the blood smears in your examples, the more difficult it would be for the algorithms to differentiate.
Yes, it's a very complicated question, because you're right. To the first order, if you have an anomalous structure on a background and they're similar, when I say similar, I'm in this situation, I'm talking about visually similar. If they're similar to your eye, then yeah, machine learning algorithms may have a problem in distinguishing them. But the interesting thing about HSI is that it does look at color, but it also looks at the underlying material. So what is being reflected back to you is not only the color, but also the quality, the physical photonic quality of the structure that you're dealing with. So there are situations where you can have a red background, like a red piece of paper, and you can drip blood on it, and still be able to distinguish objects. Again, the idea being that yeah, they're both reflecting red, but to your eye, but to the HSI camera, it's a very different type of red. There's the red liquid, and then it's the red paper. And the photonics are different. So again, I hope that answers your question or addresses your question, not question, or your comment. But I wanted to make it clear that the HSI is reflecting back basically not only color, but the quality of the material that that you're imaging. That's what actually um, the U.S. Air Force and other agencies utilize in their search for people and their search for other things. The eye in the sky, that satellites, drones have been indispensable for detecting things. That's what's very, very interesting about the technology, and we're constantly improving it. So applied optics has taken off this century, and we can do things unimaginable, things that we could never even think that we could do 50 years ago because of basically the advances in CCDs and all the techs and optics that go into basically this technology. So again, just a comment.
It's fascinating to see how much detail can be revealed in the HSI, like you said, structurally versus just what we would notice with our eyes.
Correct. That's one of the points why we use it. One wouldn't think that you could resolve certain things, like for instance, in a lot of the images, if you look at them closely, you can actually see the digital padding in some of the images. You can see basically the background digital padding on the material. And again, this is a crude piece of HSI. There are HSI cameras that can really, really resolve it in a way that you couldn't fathom. So again, this is a crude HSI. It's a 25K camera. So it's not the most expensive. The military has HSI cameras which are over like 100,000. They're really, really high resolution, and they're indispensable for doing a lot of problems. This was something that we wanted to explore to see if this tech could be utilized for helping law enforcement and outside agencies do their problems. You have a man down in the field, you want to basically really assess his situation in terms of his wound before you send your people. It's possible that an HSI/MSI camera could be helpful. Helpful maybe also with SWAT. You want to SWAT people, as we know, they use not only a shooter, but someone who's also spotting. This technology may be useful with helping spotters really assess the situation. Basically trying to do what they need to do. In this age of terrorism, it's a very significant thing to consider. Tech to really keep abreast of problems which are just exploding and we don't really have the best solutions to them.
Thank you. We have a question from the chat. Does the HSI include the ability to select combinations of excitation wavelengths with different bandpass filtration of different wavelengths and energies?
Yes, very cool question. Yes, the tech has, I hope this answers your question, but the tech has a series of bandpass filters surrounding each of I think it's over like 700 spectral bands. So it's basically, I want to make it clear that when we look at energy at a specific band, it's through the bandpass filter that's surrounding that band. So there's leakage of energy between the bands, so they're not totally independent. So it's kind of related to what you're saying. But yes, the tech basically has those bandpass filters. Again, I want to make it clear that we're sensing passive radiation. This is not the situation where we're using a laser to excite the material and then image basically that excited energy. No, this is passive room temperature energy that an object is reflecting, and the camera is just sitting there gobbling up all this energy and splitting it into spectral bands between 350 to approximately 1050 nanometers. So we're going from basically the blue, which is around like 300 nanometers, all the way to like the near infrared, which is 1050.
Excellent, thank you. How quickly does this analysis take place? Could this be used in a real-time video feed?
Yes, it can. These algorithms, very good question. These algorithms were selected because they were quick. There are other algorithms in machine learning that take a really, really long time. These algorithms are based mostly on matrix inversions and basically just numerical adding. So they're pretty quick. You program it in C++ and it will zip through it. Though you're dealing with gigabytes of data, it could be used in a real-time feed, provided that the image chips that you're using are manageable. And of course, given things like memory and all those other things. But in principle, yes, because I chose these algorithms specifically because they are quick. I didn't want something like deep learning or something like that that takes a very, very, very long time. Impractical, and it's like using an axe to deal with something that requires a scalpel. So I didn't want to do that. So great question. Thank you.
Thank you, that's very cool. Another question from the chat. Theoretically, what enhancements would be needed to visualize differences in smears versus droplets or detect differences in saturation of blood? And another comment, is there another copy of the poster available? And if Dr. Scott is amenable, we can post a PDF copy of his poster when we post the recording of this session.
Right. To answer the first question, in terms of differentiating, I think you said droplets from the background, those small anomalous structures. There's two avenues you can go down, which we talked about previously. First one, polarization, which I was talking to Rebecca about. That can sometimes, not all the times, help you basically in performing stronger image segmentation. There's another way to do it, but it's a bit impractical. That requires exciting the material with an energy source and looking at the background energy. That's another possible method, but kind of impractical. Also, another thing that people experimenting with, they're experimenting with using different bands. I'm looking only between 300 and 1,000. That's basically the visible and the short wave regions of the electromagnetic spectrum. There are people who are looking at the long wave stuff, and that's oftentimes the place where you can find some really, really cool things. I'm not looking in that region. So you can think of it like if I had like the helmet from the Predator. You all guys remember the Predator, the movie The Predator with Arnold Schwarzenegger. You remember the Predator can see in different spectral bands. Imagine having a helmet where we can basically tune our goggles, our filter, to look in a very different band, the long range. So we're looking like maybe like not heat, a little bit before heat. Heat is the infrared region. That's like 2,000 nanometers. Just come back a little bit from heat, like 1,200, 1,400 nanometers. Look in that region. Oftentimes, you'll see some really interesting things that can help you segment or differentiate between the bloodstained cloth and clean cloth. But great question.
So this technology right now, as you're imagining it would be applied, is for distance imaging. If it were to be used up close, perhaps in a field or in a handheld device with goggles or other displays, it could possibly be combined with an excitation device or other radiation source.
Correct. In principle, if it was closer, yeah. Is it easy? No, but in principle, is it possible? Yeah, it is possible. Physically, there's nothing that says that you couldn't do it. Whether it's something that you can actually do, that's a different question. But one could do it. We know, like in laboratories, we do experiments with people who work with nanostructures, there's colleagues of mine that do air-sea interaction. They oftentimes use lasers to basically ignite, so to speak, or really irradiate their source. And then they basically look at how that energy dissipates over time. That gives you an idea of what the material that you're looking at is made of, what its dynamics are, those sorts of things. So in principle, yes, you could do it. There are a lot of clever things that people never thought that they can do that they are trying to do. Putting things in a handheld device, of course, it has its own problems, but again, as things get shrunk smaller and smaller, we're finding very interesting ways to deal with it. And there's actually people at your institution who are basically investigating those things. Went to a presentation yesterday where that same issue came up, how to deal with shrinking devices and still maintaining the integrity of the device so that it can do what it needs to do. So the engineers are clever and they're coming up with ways to do things that we didn't think was possible. MSI Tech may be the way to go, because instead of using all those spectral bands, like 700, we can maybe just use like 10 and then use the energy that we could have used in those other bands only for those 10 bands. So don't use over 600 bands, use only 10, and that energy could be used to really do high resolution work for those 10 bands. So it's possible, yes.
Can you think of enhancements that might be added to differentiate between smears and droplets on a substrate, or detect differences in saturation of blood, volume of blood on a surface?
That's a harder question. We can detect abundance, but that abundance is it's a two-dimensional abundance. So we can detect I understand what you're saying, but what I can tell you is that we cannot detect volumetric blood in a way that you may be thinking, only as it appears, and I'm stressed on appears, in a 2D image. So at this point, I don't know if I'm not using those methods. There are people at RIT and other places that are doing research dedicated to optics, which are trying to look at true abundance from looking at an image in a 2D, two-dimensional, basically in a plane. But I'm not doing it. But there are methods. I can research them and get back to you, but I'm not doing them in my work.
So right now, the way this technology is being applied, you can use it to detect basically the square footage of area covered by blood, not necessarily the density or volume of the blood.
That's correct.
Excellent. Thank you very much for that clarification. Do you have any closing remarks or anything you'd like to add?
I think, I just want to thank everybody for taking the time to attend my presentation. Again, it's about giving a different perspective on applied optical technology, and also machine learning analysis, things that you may or may not be privy to. Again, I work in intelligence, but a lot of the techniques are transferable to forensics, at least I would say like the skeleton of them. I thought that this would be a pertinent project to basically share with you. And I'm very happy to take queries. I'm really, from my perspective, my work is really aimed at trying to pique the attention or prick the attention of the students about some of the cool things which are going on out there that are forensics related, that they may not have been aware of. My next two presentations which are tomorrow, are really gauged for the students to, again, to prick their attention. And again, I'm very happy to share my work. My colleague, Sarah Jensen, she couldn't be here, but she's, we were both very enthused to come. I'm sorry that we couldn't be there in person, but nevertheless, I'm glad to be able to share the work with you.
Thank you very much, Dr. Scott. I appreciate your time presenting for us here today, and I'm looking forward to your other sessions.
Thank you.